tea break, and we can move to our next speaker, Randall Wisser from INRAE.
Thank you.
You are a breeder and geneticist. Am I speaking that right?
Something like that.
Something like that. Perfect. Then the floor is yours, please.
Thank you. I also like to extend a thank you to Fred and to invitation to the meeting and to be with you all here. This is a really wonderful meeting. I think another speaker said they feel really at home. For me too, this is a great place, and I'm having excellent conversations with several people. So today I want to talk to you about some work that we've been doing to develop a uniting crop model and genomic prediction, much in the lines of work we've heard from several groups, including work Mark Cooper's group. We've been moving along this kind of similar path. And I think to just set the stage for how we discuss or think about, or how I'm trying to think about the problem we're working on is to think about crop adaptation, is to put this in a framework of not just G by E, but we often neglect the context of selection and the fact that we're dealing with population genetics that are also moving. So there's interactions not only at the scale of genetics and environment, but also the directed evolution of crops through plant breeding. I'm starting to try to refer to this as something like a GPE synthesis. Can we work towards developing a synthesis view and understanding that involves the interactions at these different scales through modeling or other strategies? And I think the time is really growing in a good way, and it's right in its place to be able to advance this kind of a framework, just because of the extensive developments, first, of course, in genomics and phenomics, understanding of plasticity by many of the physiology groups. Our understanding of diversity just continues to grow, and our capacity to characterize that diversity grows. And there's now starting to so we're starting to see more applications in understanding how these are interacting in the context of at least short-term evolution, where we're really dealing with empirical data sets, not just the theoretical side of thinking on this. And many of the people in this room, of course, are leading the way towards this type of potential integration. So with that said, I want to just explain to you a little bit about the direction of our group right now. Is very much focused on, it's been focused on flowering time. They've been working on flowering time through a number of studies, and one that that we completed and we're still generating some papers about is a population that we selected in parallel across 8 locations throughout the United States that span the latitudinal range of Wisconsin to the Caribbean island of Puerto Rico. And so what we did here is we created a synthetic population, and then this population was grown with 10,000 plants at each location, and we selected the earliest 500 genotypes, recombined them, and advanced them to the next generation of selection that derive from that specific location. So we basically have a collection of 8 lineages that were selected across all these locations, and we've published some recent studies that that have investigated what's going on in the response to selection across these populations. And I would like to note for anybody interested in germplasm, we also published a paper on the release of these 20-so populations that were developed, and they're accessible through the United States National Germplasm Program. So that's just to give you the understanding that we're working on flowering time, but after myself joining the LEPS laboratory in Montpellier, we started to embark on this interface between working towards developing a modeling system to be able to develop that synthesis. And so that's the orientation. And today I'm going to highlight the work of Arnaud Desbiez-Piat. He's a very talented postdoc in the group. I'm sad to see him go, but happy he's joining as a permanent researcher at INRAE and another group. And he's done some really nice work, and this one I want to share with you today are some of the results from that work. But essentially, what we're doing is we're using a kind of evolutionary genetic model, really a genome simulator. And the genome simulator needs to be parametrized by certain things. Recombination, we can use theoretical parameterizations, but we've chosen to try to take an empirical-based approach. So we parameterize, recombination frequencies, genetic architectures of traits that are underlying parameter traits for the crop growth model, and real genotype data. And that gets integrated to go through the genome simulator to make virtual crosses. And then the virtual crosses made from real parents are then assigned by the genomic predictions the physiological parameter traits from the genetic architectures. And then that passes to the crop growth model with the environmental data of a given environment, and those virtual genotypes then are kind of grown in a real environment, let's say. So what's going on here is we're coupling genome simulation of the polygenic traits with crop growth models. And now I'm going to just walk you through in a more practical way or practice-based way about how that's happening. What are we actually using as data, and what are the steps that this process has involved in doing this? The first is that we were relying quite a bit on a phenotyping platform where we can test about 2,000 individual pots, but so we can replicate individual genotypes several times in the platform system and extract data from the platform. And in this case today, I'm going to only be talking about a limited number of phenotypes that we pulled out of the trial data from the platform. And that would be parameters that underlie the rate of leaf appearance with thermal time. So this is the phylochron, the preformed leaf kind of intercept component, which is a bit of an abstract component, but some amount of thermal time for the first leaf appearance, and then a total leaf number. So I'm just dealing today more or less with just a few components of the system. And what we're doing is we're using the phenotyping platform to get measures of the genotype-specific values for each of these particular parameters. And then from that, we use this diversity panel, about 350 lines, as a training panel for these parameters. And so then we get the genetic architectures for those three parameters. And after we have that, we kind of embed the architectures, let's say, in a genome simulator. And so those architectures are present there. So as soon as we send we're taking real genotype data. This is genotype data that has been imputed from the diversity panel onto a collection of populations called the nested association mapping population of maize. So we're taking individual genotypes, pairs of parents, putting them into the simulator, making a virtual cross. According to recombination maps that were also estimated for these, for maize, and then generating virtual recombinants. And those virtual recombinants here are shown in this graph for a set of crosses that we decided to focus on in this particular part of the study, what I'm going to report on today. We chose these because they're dominantly affected by temperature and not photoperiod sensitivity. That can be added at another time. So we're following a pedigree structure, and making virtual recombinants, we're following a pedigree structure of the nested association mapping population, which is biparental crosses. We're making multiple different biparental crosses. These virtual recombinants get formulated by the simulator, and then the progeny of these virtual recombinants are predicted with respect to the three parameter traits. And then that's passed to a crop growth model. In our case, we're using an in-house model of serious maize and following a formalism from within that model. So this is just extracting a piece of the crop growth model to show you how those parameters are used to predict the flowering time in thermal time based on those parameters. Then we take those virtual progeny and grow them based on planting information, planting date and location, and temperature data in this case. The temperature data we're getting from NASA Power. And then finally, what that gives us is each population's distribution of the phenotypic variability in flowering time per location. What I want to do is walk you through a few results of the validation parts of this process. First is the training panel for the parameter traits. And I know other speakers have also shared similar results like this. So here's our formalism response using the phylochron and the leaf number, et cetera. And here are the individual components trained by our BLUP model, the prediction accuracies from tenfold cross-validation for each component, and then applying the formalism with those specific combinations actually, in our case, boosts the prediction accuracy up to about 0.68 here, which is quite comparable to a direct prediction on the flowering time, thermal time to flowering. It doesn't seem to matter which alphabet we go with here. We basically get very similar prediction accuracies for these traits. So the next thing is that I mentioned we make virtual recombinants. This slide's just to show you how the training panel that we're using exists with respect to the space of the real populations, as well as how the real populations overlap with one another using the real RILs based on their genotype data alone. That's all this is, just genotype data. The genotype data of the RILs, say B73 crossed with NC358. You have the blue points representing the real observed RILs and the green points representing the virtual RILs. So we get a overlap in the distribution of their space. And then if we take and we project the genetic architectures, pass them through the formalism, and then grow these genotypes, these virtual genotypes, in one of the real environments where the NAM population was grown, such as here in New York, 2007, you can see the distribution of the real RILs in terms of the phenotypic variation of flowering time, and in days now, and the virtual RILs. And we have quite a nice overlap in those distributions. And then here, if we just look across more environments, you can again see per population. So this is one of the populations we simulated, another one. So populations are here, and environments are in the columns. And then we have the predicted in red and the observed in blue. And overall, from looking at these particular pops and environments, we get a mean bias of -1 to -4 calendar days to flowering that are predicted from these digital recombinants. So that's telling us the system works pretty well at predicting the timing of flowering, as well as fair good overlap in the distribution that we observe. So now, in the last few slides, I'm going to switch gears and just talk about how we then move to upscaling that beyond the validation stage, and just now we go to sending these virtual recombinants into many other environments and what we might be able to learn from that. And what I want to say here is that we turn our focus, when we think about the variation in flowering time that's expressed in days, we like to think about it as the variation from the first individual to flower in the field to the last individual, as opposed to summarizing the variation that from the planting date to the last date. So all of this shared history of growth and temperature doesn't kind of accumulate into error later on. So our decision was to try to focus on the flowering window only in describing what's going on in this interaction with temperature. And what happens here, as you can see in this plot, is when you focus on this flowering window, and in this environment you have a cold spell that occurs during the flowering window, that alters the phenotypic variability of the population, where it shows a kind of bimodal distribution. And if you go into another environment and you have a hot spell occurring during the flowering window, this leads to a more sort of normal distribution. But a key thing to notice is how these two distributions are either expanded or compressed by that temperature. And I think there was a question earlier in one of the talks when Danielle was talking about how does G by E arise from differences in scale versus differences in rank. This is scale-based variance change. So this is that component. This is showing you how temperature can give rise to compression or an expansion of the phenotypic variance of the population that's environmentally dependent. It's not rank change. There's no rank change G by E going on here. So now, if we take we scaled up the number of crosses. We made 10 crosses, 200 RILs each. So we have 2,000 RILs that were grown in 2,000 real environments, 2,000 virtual RILs and 2,000 real environments. And then what we looked at was what Arnaud looked at was the average temperature of each environment in that flowering window, and related that to the within environment heritability of these 2,000 RILs. And what you can see is as we go from cool temperatures to warm temperatures, the heritability undergoes a substantial drop. And that's because of this compression of the variability. So as the phenotypic variability of the flowering window compresses, then we have a reduction in the heritability. And so in this case, we can see this, we made some assumptions about the residual error in this case, and it basically takes what is otherwise a logarithmic decay and converts into a linear response, where we see a 40-point reduction, 40-point range in heritability across this temperate zone of environments we analyzed, with an average of a 3-point reduction in heritability per degree change. And about here, about 32 is really the fastest rate of development that you would get, and so the compression at 32 starts to get to the bottom level. And so as environments in the future move from where many of them are today, a few degrees later, it's going to pull down those heritabilities. At least that's what the model would project. And so that raises some questions about how breeding progress itself will be affected under climate change. So what I'm going to summarize and let you know that I think that by interfacing physiology and quantitative genetics, for me, has been a critical eye-opening link to be able to think about this GPE synthesis that I mentioned. There are several validation challenges that we encounter in the process of this. In particular, at what granularity of G by E can be predicted. For instance, there's existing error in the real data itself, where standard errors can be sometimes can get large, and it leads to a lot of uncertainty about whether your model is right or whether the real data are right. Offset, we tried to look at, what I didn't talk about, we tried to look at if this effect on heritability could be offset by other means. For instance, the breeder's equation, we found that it requires almost a doubling of the selection intensity, which ultimately would just lead to a reduction in diversity overall. It may affect the longer-term response to selection. We expect that theoretically. And management, we tried looking at the shifting of management dates by as much as a month, and it makes no difference doing this. You can't overcome this particular temperature impact. So interfacing, coupling, and integrating, all the things we were talking about this meeting, I think it's a great new way to think about and guide strategies for adaptation. We can extend this system and develop it, and it helps us think and test counterfactual scenarios that we can't otherwise do in the real world, as long as we can get these models to reflect reality well. Lastly, in closing, we have a new postdoc opportunity. We have a new project called Fusions. It's a partnership with a group in Paris, Les Moulins, and basically we're now extending this out to the selection framework and the evolution framework. And so please contact me if you know any postdocs interested in this position, or if you're here in the audience, come talk to me. And thank you to many people who have helped along the way. Yeah.
Oh, that was absolutely amazing.
Okay.
Unfortunately, we don't have much of time for the questions. Sorry. Are you trying to avoid those guys?